Thanks to internet pop culture, I'm sure we've all heard the phrase, mitochondria are the powerhouse of the cell, at least once or twice. Cells are the small subunits that make up all living things. And just like our relatively large and very complex bodies are divided into different areas of specialization, which we call organs, our cells are also divided into areas of specialization called organelles. The mitochondria are the organelle that produce most of the energy our cells need in order to function, hence the powerhouse of the cell. Mitochondria are interesting for a variety of different reasons, one of which being that they have their own DNA. DNA are the blueprints that our cells need in order to function, and damage to mitochondria or their DNA can lead to a variety of diseases which affect organs all throughout the body, some of which are listed behind me. The heart and the brain are two organs most commonly affected by mitochondrial damage, which makes sense because these organs require a lot of energy in order to function. As an undergraduate, I was part of a team interested in studying mitochondrial DNA transcription. Now, we can think of a cell as a house, composed of many different rooms with a variety of different purposes. The DNA, or the blueprints for this house, can be rather difficult and confusing to understand. So we need an interpreter of some kind, an architect. In this case, cells use RNA. This RNA tells us exactly what we need to build and where we're going to put it. How many bedrooms and bathrooms are we going to have, and where does the kitchen go? It's much easier for us to study this RNA than it is for us to study the proteins that actually make up the cell. And by studying the RNA, we can figure out what a cell is actually doing, instead of just what it's capable of doing, which is what we would find out from studying the DNA. In order to do this, we need to be able to actually look at the DNA and RNA that is present. So as an undergraduate, my job was to develop a process to use fluorescent dyes to look at mitochondrial DNA. This is an example of one of the tests that I would actually use in my research, and is in fact a sample of the tests that I ran. This is one of the pictures that I took for publication. These are all treated with a fluorescent dye, so they're samples of RNA and DNA. And the way this works is this dye is present in the solution with the RNA and the DNA. We shine a light on it and it glows. And we can measure how much it glows or fluoresces. On the far left, you'll notice that we have one column. We call them lanes, and it has multiple different bright areas. These are different pieces of DNA. We know how much DNA is present in each one of these bright areas, and we can compare that with how much they fluoresce, and then use that to compare against our samples where we don't know how much DNA is present, and calculate how much DNA is present in all of our samples. We want to use this technique in order to study how different factors affect the production of RNA from DNA, and hopefully one day develop techniques that can help create therapies for those mitochondrial diseases.